The Chinese alligator is the first genome of its kind to be sequenced. What we learned from sequencing this genome can help in preserving this endangered animal and other crocodilians. This study focuses on the diving and sensory properties of these organisms. The most notable discovery of this genome is the fact that crocodilians have no sex chromosome. So studying this genome can help in determining how sex chromosomes evolved over time, but more on that later. The molecular landscape of the crocodilian's genome is very unique. Crocodilians have the highest guanine cytosine content of all sequenced genomes. Not only that, but the GC content is very evenly distributed throughout the genome. This makes the DNA very stable and can contribute to the fact that crocodilians have existed for such a long period of time. Further segmentation analysis also suggests a very close relationship to chickens.
The researchers employed tree farm to deduce gene clusters within 5 reptiles, being the Chinese alligator, the green anole, chicken, turkey, and zebrafinch, and used humans as the outgroup species. With the data exhibited in Figure A, the researchers were able to conclude that the Chinese alligator developed more unique paralogs and unclustered genes, as well as had more lineage-specific genomic features. From the unique paralogs determined, the researchers were able to identify 413 alligator-specific gene families. Based on the single-copy gene families of the species under study, the researchers were able to construct a phylogenetic tree and calculate divergence using fossil fuels. This revealed that the crocodilian lineage split from the common ancestor of birds approximately 241 million years ago. In addition to an expansion in paralogs, adaptation features of genome divergence induce an excess of non-synonymous over synonymous substitutions at orthologous genes, which can be identified as positively selected genes, PSGs, by the likelihood ratio test and lineage-specific accelerated gene ontology categories by the binomial test. Using these tests, researchers identified 219 PSGs and 86 evolving gene ontology classes. This data proves that rapid evolution was occurring within the species under study.
Positively selected genes of the alligator are largely metabolism related, which suggests a special energy demand for diving. 24 of these 28 genes are related to oxidative phosphorylation, the director of energy production. 2 ATP synthases, which catalyze energy production, have also undergone positive selection. Because gas exchange is impossible during diving, the route of excretion is via sodium bicarbonate in the urine. The transporters for these ions are overrepresented, and PSG-enriched pathways generate their precursors.
The Chinese alligator has many unique oxygen pathways. 3 of the 4 crocodilian-specific hemoglobin genes are bicarbonate binding units that promote oxygen release to tissues. They also possess routine phosphate binding and active oxygen transport. During submergence, the alligators slow their heart rate and supply oxygenated blood only to the brain to adjust to oxygen depletion. Gene ontology reports show a rapid evolution in diving hypoxia adaptations. They minimize their metabolic rate during diving through a longer resting rate and slower heartbeat of robust cardiac muscle. Their genome shows evidence of excellent olfactory, auditory, and ocular systems. It possesses the most abundant functional gamma and alpha olfactory receptors, which bind airborne and water-soluble odorant molecules. Opsin for dark adaptation, autopetrin for underwater sun detection also evolved rapidly. Their genome features antigen-triggered adaptive immunity and strong innate immunity. Also, the overrepresentation of cathelicidin, a major cause of antimicrobial peptide in the blood, shows an effective system of defense against microbial infection.
Usually, comparing expression profiles reveal major differences between species, but the pairwise comparison of the genes involved in sex determination in the Chinese alligator and chickens only reveals slight differences, suggesting that the genes in the Chinese alligator may resemble the ZW system of the chicken. The M-fish results suggest the alligator chromosome 3 and chicken chromosome Z share an ancestral chromosome. This shows that the sex chromosomes are not necessarily needed to determine sex. The researchers looked to DMRT1. Aligning this alternatively spliced variant of DMRT1 revealed testes-specific isoforms with only the DM domain, and none of the ovary-specific isoforms had DM as their only domain. This suggests DM bias is necessary for genital expression profile in Chinese alligators. The DMZ isoform likely plays an important role in sex determination because it is the only isoform that can have DM as its only domain and because it is very similar to the chicken DMRT1 V4 isomer. Analysis of the SNP showed very low heterozygosity rates and therefore low genetic variability, suggesting that Chinese alligators have suffered from a severe bottleneck in the past. Using the SNP data in the PSMC model, we can deduce a rapid increase in the alligator population between 0.6 and 1.05 million years ago. Change in the region of 0.6 to 1.1 million years ago explains a mixing of populations.
This sequenced genome, the first complete crocodilian genome, shows evidence of terrestrial-style reptiles adapting to aquatic environments and is a valuable resource for designing strategies to protect this endangered species.